Bismillahirrahmanirrahim. Today we will know what is T-ARMS-PCR and how it works. We will know about the T-ARMS-PCR and the working mechanism of the T-ARMS-PCR, that how it works. The T-ARMS-PCR actually means Tetra Peimer Amplification Refractory Mutation System PCR, meaning that it has 4 primers in use. Actually, it is a type of PCR which amplifies a specific DNA sequence like given in this figure. This PCR will amplify this specific given sequence. It means that this PCR works only when there is the presence or absence of a certain nucleotide. For example, in this figure we see that there are two nucleotides A and T present at a specific position. If we design a T-ARMS-PCR for this nucleotide sequence, then that primer will only work when the T nucleotide is present at this location, and when T nucleotide thymine is not present at this location, then the primer we have designed will not work. So this is the specificity of the T-ARMS-PCR. What is the principle of T-ARMS-PCR? The first one is that it exploits the inability of take DNA polymerase to extend primers mismatch at their 3' end. If the primer we have designed for a nucleotide sequence is not complementary and is not matching to the other template sequence, and there is a mismatch at the end of that primer, then the DNA tech polymerase does not have the ability to extend the primer and carry out the polymerization reaction. So this principle is used in T-ARMS-PCR, that we introduce a mismatch at the end of the primer, and when this mismatch is present, the DNA polymerization will not occur and their sequence will not be amplified. Let's see. The second principle of the T-ARMS-PCR is the introduction of a second mismatch, which further destabilizes the mismatch primer and hence specificity is increased. Further, we want to increase the specificity of the T-ARMS-PCR, then we introduce a second mismatch that is at position number 3rd of the primer end, that further decrease the binding of the mismatched DNA sequence primer to the DNA sequence. If there are two mismatches, then there is very less possibility of that the primer will be extended and DNA polymerization will not occur, and mostly if there are two mismatches, the DNA polymerization will not occur. And the third principle is that the DNA polymerase tech DNA polymerase does not have the ability to repair and exonuclease activity and remove mismatches. The tech DNA polymerase cannot remove the mismatches. For example, we have the mismatches that we have introduced at the 3' end of the primer will not be removed by this DNA tech polymerase, and the extension will be stopped. What is T-ARMS-PCR carried out for? T-ARMS-PCR is carried out for finding the different nucleotide present in the DNA sample of different individuals, of a different species. As we know that in a population there is variation in the genome, variation in the gene, and variation present at the nucleotide level. For example, we have a gene that has a nucleotide A at a certain position in individual A. The second individual has a G nucleotide at that position, and the third individual has a nucleotide T at that position. So if we want to know which nucleotide is present in the individual we are studying, or the population of individual we are studying, then we will design T-ARMS-PCR primers that are specific to these nucleotides. One primer will be specific to A nucleotide, the second primer will be specific to the G nucleotide, and the third primer will be specific to the T nucleotide. We can design primers specific to every nucleotide. So on the basis of that, we will be able to find out that the individual is having which nucleotide, A, G or T. So this is basically T-ARMS-PCR is carried out for. What is an SNP? SNP what means single nucleotide polymorphism. It means the existence of different nucleotides at a certain position in a gene in different individuals. Suppose we have three individuals that have three different nucleotides in a gene, then we can say that their nucleotide is this is single nucleotide polymorphism. As we can see, this are DNA sequences from two individuals. These two figures shows DNA sequences from two individuals. The one individual has a C nucleotide and the other individual has a T nucleotide at these positions that are pointed out by the arrows. So this is the variation called single nucleotide polymorphism in genes. Actually, this gene is the microRNA 585 microRNA gene, which is different in different individuals. Some individuals have C nucleotides and some individuals have T nucleotides at this position as shown in the figure. We can see that there is a presence of C and G in one individual. This individual possesses a single nucleotide C at this position number 11 or position number 9. We can see there the variation by zooming in that there is a presence of C nucleotide at this position. This is the DNA sequence from a second individual, and we can see that this nucleotide, by zooming in, we can see that this individual has a T nucleotide at that position number 9. So these two individuals have different nucleotides at that position number 9 in this microRNA 585 microRNA gene. So these individuals are different, have different alleles of these genes, and we will use T-ARMS-PCR to find out which nucleotides is present in the individual that we are studying or the population that we are studying. This figure shows the DNA sequences of two individuals having different nucleotides at this certain position. This is the DNA sequence of these two individuals, which differs at a certain point. So these individuals have two different alleles for the microRNA 585 gene. Now we should find out that why we want to carry out a T-ARMS-PCR. It is simple, it is repeatable, and it is economical. It It is used to find out the frequency of an allele in a given population. Suppose we are studying a population of 500 individuals. Then it tells us that how many individuals have C allele, how many individuals have the T allele, and how many individuals have the C and T allele both present as heterozygotes. So it tells us about the allele frequency of a certain gene in a population. Later we use this frequency, the presence or absence of semi-nucleotides, for associating this nucleotide polymorphism with a disease like coronary artery disease, cancers, diabetes, and other diseases that can be affected by the that can be studied with the T-ARMS-PCR, and the disease may be associated with the single nucleotide polymorphism. Or it can also be a protective allele. We can also discover a protective allele against a certain population condition or disease like lung cancer, breast cancer, etc. If we consider a disease in that perspective, then we can find out that the presence of a certain allele in a population is able to protect that population from developing a certain disease like lung cancer, breast cancer, liver cancer, diabetes, etc. T-ARMS-PCR provides an easy, reliable, and cost-effective method for finding frequency of a single nucleotide variants in a given sample size. So these are the points that we use T-ARMS-PCR. Now let us study how a T-ARMS-PCR works. In a T-ARMS-PCR we are using 4 primers. Okay. Of these 4 primers, the 2 primers are very unique and different from the conventional PCR primers, and these 2 primers are called NR primers. The 4 primer includes these are NR forward and NR reverse primers. The NR primer are NR forward and NR reverse 2 primers, while the other 2 non-specific generalized PCR primers are known as the outer primers. These are also 2 in outer forward and outer reverse. So we use 4 primers in a T-ARMS-PCR, 2 NR, 2 outer. The NR primers have forward and reverse primer, and the outer primers have primer and reverse primers. Now let us consider the example of the human microRNA 585 gene, which we have discussed earlier. Now let us design primer for that gene and see how it works. This is the microRNA 585 gene in an individual 1. Now we take another individual that also have the same gene but a different nucleotide at this position. We can see from the arrows that these 2 individuals have 2 different nucleotides at this point that are highlighted in red color. One individual has C allele and the other has T allele. So these are the variants from the microRNA 585 gene in humans. The variants are C and T in 2 individuals. Now we will design primers for the 2 variant alleles. We will design a primer that is able to amplify the C allele. Similarly, we will design a primer that is able to amplify the T allele. This we have designed 4 primers, of which the 2 NR primers are given. We can see in these figures. One primer is the forward NR primer that is specific to the T allele. This primer will only work when there is a T allele present in the genome of the individual in the microRNA 585 gene. And when this primer works, it will give a product whose length is 202 base pairs. We have a second inner reverse primer that is specific to the presence of a C allele in individual genome in the microRNA 585 gene in this position. And the second primer, reverse primer, will only work if there is presence of C allele in the individual. If C allele is not present at this position in the genome of this individual, then this primer will not work. If there is presence of C allele in this individual and the microRNA 585 gene at this position, then this primer will work. And when this primer works, the product formed by this primer will be 295 base pairs. On the basis of this product length, we will able to identify which primer has worked and which primer has not worked. By comparing the length of this primer to the DNA letter or the DNA letter, then we can see which primer has worked. If it's the product length of the product is about 200 to 200, then we can see that the primer inner forward as primer has worked, and if there is then the T allele is present in the individual. The product length of the PCR is about 300, nearly 300 or 297, then we will see that the reverse primer has worked and the individual has C allele in this gene present at that position. There are also two outer primers. These are, we have designed 4 primers. The 2 inner we have discussed in the previous slide, and these are the 2 outer primers. When these 2 primer outers work, they will form a product of base on 446 base pair. This will be the product of the outer primers, and this will be present at the base of the agarose. When we gel the agarose, in the agarose gel, then we will find this product at the base of the line. So these 2 are the outer primers. These are general PCR primers, and they have nothing more special than general PCR primers. Let's see in this figure that how the primer gets attached to the DNA sequence, to the respective DNA sequence. Here we can see that this is the primer which has a G nucleotide at the end, and this sequence is genome of the individual having C allele in its genome and the microRNA 585 gene. First, there is denaturation of this DNA strands. When the DNA strands are denatured, it will be separated into 2 strands. The DNA strand is separated into 2 strands. Now the primer will come and get attached to the DNA strand. This is the primer comes and attached to the DNA strand, and G binds with C. This G is complementary to C, and the binding occurs. When the binding occurs, then we can see that the individual has C allele, and the product will be formed, and we can recognize that product on the basis of length, of its length. Now the second individual is DNA is denatured, and the 2 strands will get separated. Now the 2 strands get separated, then another primer will come and try to attach it. This is another primer that comes and get attached to this DNA sequence. As this DNA sequence is from another individual, the other primer is able to get attached to it, because AT is complementary to A. We can see that these 2 primers are the inner primers, and these are allele specific primers. Allele specific means that these primer will, the first primer will get attached only if there is the presence of C allele, and the secret primer will only get attached when there is presence of TLE. Now let's further see its details. This is a DNA sequence from an individual having the allele T. This is the complementary strand update DNA. With these two strands are separated, a primer will come and get attached to it. This primer is called a forward inner primer. The primer that is shown by the arrow is a forward inner primer. This is another DNA sequence. This is the complementary strand of the DNA sequence. A primer comes and attached to the forward strand, in which the end of the primer G is complementary to the C variant and the forward strand. We should keep in mind that the forward primer gets attached to the reverse strand because it will make the forward strand on the basis of the reverse strand, because the DNA polymerase enzyme will read the reverse strand and make the forward strand by adding complementary nucleotides to that reverse strand. And the reverse primer will get attached to the forward strand, and then it will be extended to form the reverse strand, which is complementary to the forward strand. So this is the reverse primer, inner primer that gets attached to the forward strand to make the complementary reverse strand by reading the forward strand. The inner forward primer attached to the reverse strand of DNA only if there is C allele or nucleotide at this position. Otherwise, it will not get attached. We can see in the figure that this is the first is the nucleotide sequence of a gene, which is complementary to another strand. The both strands of DNA are contained in this figure, and the other figure is that of the primer that tries to get attached. We can see that the G is complementary to C and it binds. The G is complementary to second G is also complementary to the nucleotide C and it binds, and the T is just cannot bind to the as conventional glue T cannot bind to the C. So this is the mismatch we have introduced in the primer that is specifically designed to increase the specificity of the reaction. The other nucleotides also gets attached, and this primer will be extended and the polymerization will occur. So this is a primer that is attached to the nucleotides to the gene microRNA 585 gene. The nucleotides that have been included in the yellow circle indicates the mismatch introduced intentionally. That is intentionally introduced or designed by the software so that there is presence of mismatch at this position. When there is this first mismatch at the first nucleotide I between G and C, that is G and C, in this position, in this case it is not present. But when there is the presence of a second mismatch at the first position of the primer, then the primer is not able to bind and the DNA polymerization does not occur. The arrow points to the intentionally introduced mismatch to increase specificity of the primer. This mismatch increases the specificity of the primer. This it means that if the G is not binding to the C, it is mismatched, but there may be a possibility that the primer will be extended having a single mismatch. But when we introduce a second mismatch, then there is very least or impossible to that the DNA polymerization will occur. So we want to remove false positive results completely, and for that we introduce second mismatch. The first arrow points to the allele specific to the C allele. Now in this figure we can see that there are two mismatches. The one that we have intentionally introduced, and the other that the primer has, because this primer is not designed for the amplification of a gene that have T allele and complementary and opposite side of this end of the primer. So when this in this situation occurs, the primer will not get extended because the nucleotides are not matching, and the polymerization will not occur, and it means that the C allele is not present in this individual, and this individual has a T allele. In the second figure we can see that another primer that we have designed for the C allele is able to bind the DNA sequence. That this primer, we can see that this primer is not able to bind in the first figure to the T allele, but the same primer is able to bind another nucleotide sequence, the of the microRNA 585 gene that has a C allele at that point. So the primer we have designed for that C allele is able to bind, and the intentionally mismatch will not stop this DNA polymerase, DNA polymerase final enzyme from polymerizing or extended the DNA strain, and the product will be formed. So in this occurs that this tells us that the genome from this individual has the C allele, and the product we will see will be complementary to the C allele primer. Now let us illustrate that how does this binding occurs. In the first figure binding will not occur, and in the second figure binding will occur. We can see that the tick mark indicates that G and C are complementary alleles, and G binds with C in the second figure. But at the first point we can see that T is not complementary to G, and the primer is not able to bind. At this point this mismatching, we can see that there is a second mismatch that we have introduced that is not able to bind to the C allele on the genome of that individual, and this intentionally introduced allele is also not working in the second individual genome, that is shown by the by the cross signs. The other nucleotides of these two individuals are matching with the primer and are shown by a tick mark that the binding between these nucleotides occurs. But in the first case there are two mismatches, and the primer will not get extended, and in the second figure we can see that the end of the primer is complementary and is working, and the only mismatch present at nucleotide number 3 will not be able to stop this primer from extending the DNA or polymerizing the DNA sequence. The red cross shows that this primer will not work, and this tick mark shows that this primer will work and DNA will be polymerized, and we will identify at by performing gel electrophoresis from the length of its product by comparing it to a DNA ladder. What are the application of the T-ARMS-PCR? We can use it for case control studies. Case control studies mean that if we want to analyze breast cancer in a population, in a certain population, then we will take about 500,000, 2,000 or 3,000 patients DNA sample. We will take their blood, we will collect DNA from their blood. Similarly, we will take healthy sample from that population and about the same size as the patients, and then we will carry out T-ARMS-PCR in these individuals for a certain gene, like that gene we have mentioned as micron 585 gene, and we will see that how many individuals in the healthy persons have a T nucleotides and how many individuals in the diseased person have a T nucleotide. Then we will compare whether this allele frequency is greater in the diseased individuals or it is greater in the healthy individual. And the difference between these two alleles in these two populations is significant. Then we can say that this allele is associated with a certain disease. It is also used for genetic mapping, and it is used for disease association studies. It is used for molecular breeding and genome-wide association studies. When we carry out the T-ARMS-PCR, then we will take this PCR product and run it on agarose gel, 2% or 3% agarose gel, and we will run on that sample and agarose gel. We will obtain a certain binding bands on the agarose gel. There in the figure we have shown that the bending pattern of the agarose gel. Either there will be presence of 3 bands or 2 bands. We will recognize these bands on the basis of the length of their product. The first band that is shown in the red color is the product of the 2 outer primers. The second band that is shown in the yellow color is the product of the primer that is specific to the T allele, and the third band that has a greenish color is the product of the primer design specific to the C allele. So in the first line, if there are presence of 3 bands, that means that outer primer is present and the primer specific to the T and C allele both have worked. It means that both the alleles are present in this individual DNA and its genotype is C and T, meaning that this individual have both C and T alleles in his genome. And the second or in the middle bands, we can see that the presence of the product of the outer band, that means that PCR is working, and the presence of the allele C band. It means that this individual has contained only the C allele and the T allele is not present in individual, and his genotype is CC genotype. Homozygous, it means that this individual is homozygous for the CC allele and on the microRNA 585 gene. And in the right 2 band, we can see that the outer right band is the band that is not specific and generalized primer, and the second yellow cell we can is specific to the T allele. It means that this individual does not have the C allele, and he or she has only T allele in his genome, and he or she is homozygous for the T allele in this micron A585 gene. So in this way we will get the genotypes of the individuals based on the length of the product form and the agarose gel. This is the heterozygous and this is the homozygous C allele, and the right hand side is the homozygous TT allele. So this is the way the gel after gel electrophoresis, the bands in the PCR gel are analyzed and the genotypes are obtained. In this figure we can see that we have a DNA marker that we will run on agarose gel, and that DNA marker will appear like this. Our primers, we have designed the outer primer length of 446 base pair, so we can see that this primer will be near to the 500 band of the DNA ladder. Our second middle primer product, which is 297 base pair shown in yellow color, will be somewhere near to 300 base pair of the gene ladder, and the greenish band which is 200, and that the band will not be greenish in the PCR gel, but we have shown in this figure for pieces in different colors. The band that we have shown in greenish color will be about 202 base pair, and we will be able to recognize it by comparing it to the DNA ladder that is shown in the figure, and it will be somewhere near to 200 base pair on the gel. So by comparing this length of these primers of the product, we will be able to see which primer is working and which is not working. For example, in the first pattern that is on the right hand side, there are two bands, the red color and the green, the yellow color, the red color and the yellow color. Then we can say that one primer is working that is specific to the T allele. And the other primer is not working, meaning the C allele is not present in this individual. In the middle bands, we can see that the presence of an outer 446 base pair product and the presence of 202 base pair product, and we know that this 200 base pair product is the result of the PCR primer that was specific for the C allele. So we can say that this individual have CC genotype. And on the third pattern of bands that have three bands present in the left corner, we can see that the presence of outer primer, the middle, and the greenish primer. So the yellow primer, we can the primer that have a size of 297 base pair, we are able to find that this is the product of the primer that was specific for the T allele, and we can see that this individual has a T allele. But on the other hand, this individual has also another vein that has a size of about 202 base pair, and we know that this is the product of the primer that was specific to the C allele. So in this case, we can see that this individual have both C and T alleles, and that is a heterozygous individual, and his genotype or her genotype is CNT, CT, mean that he or she has genes of both alleles from different parents. So this is how the PCR bands look like in the agarose gel, and this is how we will recognize them by comparing their size to the DNA marker that we are running on the gel. And also important that when we run a single gel, then we are able to recognize these bands without running a marker on the basis of our previous experience. So this is further illustration of the PCR that how it works. This figure shows the DNA sequence and the two outer primers attached to this DNA sequence. The second figure shows the inner reverse primer and the forward outer primer attached. The forward outer and the inner reverse primer get attached to the DNA sequence. And the third figure shows the inner forward and outer reverse primers attached to a DNA sequence. Thank you for watching this video. For further videos, please subscribe to our channel Curious Minds to get more knowledge about biology, biotechnology, biochemistry and general knowledge. Thank you.